Right, I think we're ready to go. So thanks everybody for sticking around, and I hope you're looking forward to the end of the day and the drinks, and because I certainly am. So for those who don't know me, I'm Richard Milne. I'm an ethicist and social scientist by background, and I actually work just across the road in Wellcome Connected Science, where I lead a team of researchers who work on kind of emerging ethical questions associated with developments in genomics and biodata, which is kind of broadly the framing for this session around responsible AI. And so I'm not going to introduce the panel because you've already been introduced to everybody, but just to say that Mariana made the fatal mistake of mentioning the ethics word in her talk. And I thought, well, and has then very graciously agreed to join the panel. So thank you very much.
Thank you very much.
And I know, so we did do introductions, but I just wanted to say if there's a kind of chance for anybody on the panel to maybe say anything else about themselves that wasn't captured in the kind of, the little blurb that the various chairs read out. I know, Miha, when you talked this morning, you were talking about that kind of importance in your work of, kind of giving back to patients and your kind of your background. And I suppose I'm kind of interested in starting with this conversation, which is partly about, I think, kind of humanizing AI to maybe humanize the panel a bit beyond affiliations. Mm. So, Miha, is there anything that you want to say about how did you get here?
So for us, right, we realized how hard it is to model high-dimensional gene expression data and then to interpret these models like Maria mentioned. I think you did a fantastic job, right, of narrowing down the hypothesis space to 40 or so genes. But when you work with that many features, it's really the interpretation gets really difficult. We learned that the hard way. So what we do today to make our models better interpretable and more understandable, closer to the people, right, and to the doctors we work with, is that we infer biology phenotypes. So in the end, to classify patients in different subtypes or for response, we only work with a handful of features, right? One feature for each biology. And that, I think, makes it a lot easier to explain and to present to the public and just open up the black box that we've built. So to us, it's a lot about feature reduction, and that I think is a big aspect of what we do.
Yeah. And then moving into that, to that kind of interesting that point of explaining to the public, or like who's the audience,
who's the stakeholder.
How about you, Mariana? Who do you see when you're? I mean, I suppose this kind of gets to this idea of explainable AI. When you're thinking about explainability, who do you see yourself explaining to?
Well, first, I think it's important to say that I'm sitting in a hospital in the Brazilian National Cancer Institute, and when we are thinking about those methodologies, especially when applied to the clinics, I think there is, in general, how do I say, they don't give it credit enough. I mean, they are skeptical about those models, right? And to unbox the box, I think it's a way to show like more credibility of what has been done, and also to trust more and to be more comfortable of taking decisions based on what you are seeing. So when we talk to physicians, clinicians about these tools and how they can empower them to take those decisions, they don't understand what you mean by this, right? So if you can somehow show the biology behind, I think it's easiest way to show the value of what you are building. So I think at least for me, it's very nice to understand also with the machine, like how the biology is behind. I mean, when the machine is selecting those features, it's very nice to understand why they are important, right? What are the role, I mean, in my case, in the tumor progression. And this is also a way to show for people the value of the model per se. So yeah, I can see through these two lenses.
Yeah, it's like kind of being in that kind of middle space, I suppose.
Yeah, right.
And Philip, so you work in a kind of slightly different context, I suppose. So how do you see that kind of question of explainability in your context? I think you kind of started to touch on it with Ronnie's question at the end there. You wanted to elaborate?
Yeah, maybe we can just elaborate a little bit on that. Right? I mean, so we're in the molecular world, right? Modeling used to be very physics driven, right? And that's sort of intuitive, sort of by nature, right? I guess, right? Because you have sort of a physical intuition between what the molecules do and I think in the future, right? There's going to be maybe a bit of a merging or a bit of a convergence of these physics-based methods and these fully data-driven methods, right? As I mentioned, right, there's I do think a reasonable claim can be made that many of the like structure prediction or structure design methods learn something akin to an energy, right? Which is a physical property, right? And that can be made in the future. Maybe that can be made a bit more explicit, right? I mean, I think that there hasn't been, as I mentioned, right? The field hasn't been very focused on explainability, right? The field has very much had an engineering mindset, right? We just need to get it to work, right? And I think maybe in the future there's going to be a bit of a shift towards that.
And Stein, is that something similarly that you're that kind of moving from that mindset of we need to get this to work to how do we explain it, how do we interpret it? Is that something that you're seeing in your work?
Yes, it's quite similar in the sense that we work on fundamental problems and want to know how things work. In our case, on the genome, and so one could consider AI models that make a prediction in the clinic to be also explainable to the doctor to kind of know what it works. But for us, it's like only that. So we don't so far we don't really need a lot of models to predict gene expression because we measure it. So it is the whole point. I think for protein structure is still a bit different because you want to design a protein to actually use it. For the DNA also actually we start using that. Or maybe there are similarities and differences. But in any case, the model, when the model predicts gene expression or transcription factor binding accurately, we really want to use the full spectrum of methods to get the explanations out to gain biological insight. And so what I see is that computational biologists are really doing experiments on the model. All this, a little bit like genetics. So we would go into the lab, do an experiment, do the next experiment, try to think, do the next experiment. Here, to me, it's similar. The training the model is somehow challenging, but it's not even half the time or the resources spent in our daily research. Once the model is there, then actually most of the work is trying to use this model as a tool to get to some new insights. And so it's a particular way of working, I think, where computational biologists really have to think about experiments, about controls, all of this while training new models, testing the models, exploiting the models. So it's really fascinating. And for the regulatory genome, so I've been working on this since my PhD when the human genome came out, and with always small increases in what we could understand. And I think with many of us here, we are not here because of a hype or something. It's like really every day. Your talk also was like surprised and amazed by the performances of these things. So at the moment that we were using the convolutional neural networks, the amount of insight we got per day or per PhD student was just a lot higher, and the accuracies are unbelievable sometimes. And so that is very, very encouraging, and that's why I think we're still doing this. We might think that, I'm still thinking that this is a transition phase, that these black box models are now tedious to decipher and so on, and that they will be very useful to go back to first principles and to exploit these models to get to the actual rules. That is what I always thought, but this period is really lasting quite long, and we are still reaping the fruits of the black box models every day. So as long as we can get new information from it, I think we'd keep going for a little while.
Yeah. Yes, I did have a moment when you kind of put up the chicken genomes that you were going to unravel. Was Andre's black box pigeon this morning? So
That was
closing the loop of the day.
That was a beautiful moment today. The pigeon.
I mean, I think because that touches on something I think that you mentioned, Philip, in your talk, which was about this kind of speed and scale of progress. And I suppose there are. I'd love to hear from kind of the panel about what do you see as being the real opportunities for kind of biology over the next 10 years in terms of that, but also what are the challenges in terms of how we make best use of them, make responsible use of them, get the most societal benefit, I suppose, from those. I can go back, unless anybody wants to jump in particularly, I can go back down the panel again, but it's,
Yeah, it's good. To us, it's not the time it takes to build a model. Like you said, we get insights daily, right? And we discover so many new things and get models with great potential, but then validating this model and showing efficacy first retrospectively, and then doing the analytical validation, and then doing prospective validation in clinical trials. So getting these models to the patients takes a very long time today. And I understand why the safeguards need to be in place, right? But on the other hand, it's also an obstacle, and it's slowing down the progress in this area significantly.
Yeah, I think the volume of data we have already, it's huge, right? And I'm not sure if we have explored this data at the fully potential. So I see this as a big opportunity for, going deep down in the biology and understanding mechanisms that we have no idea that happen. Right now with the possibility of also using those LMM models to understand how molecules interconnect in the cell, I think this is an open field, and there are some works on this that, they're fascinating. And in terms of health per se, I think we are going towards a period where everything's going to be so connected. And as we saw this morning, instead of maybe treating diseases, we are going to be more at the side of preventing diseases, because I think with this power of AI, prediction is going to be everything, right? So yeah, I'm very excited about what is to come. So.
Actually, yeah, maybe one point to all the young people in the audience, and I see some of you. So I think this is really one of the most exciting times to be doing a PhD, or to be doing research like down at the front, right? And I think it's really, I think you guys are in a very lucky situation, I think, right? I think this is an exciting time. This is something that we always wanted to do, right? But we never could, right? When I did my PhD, we had to do sort of the slow trudging sort of research, right? And you get to do a lot of the exciting stuff. So I think I just wanted to make that point just for all the young people. So in terms of impact and for the near or the medium term future, right? So on the molecular protein side, right? it feels like that things are going to mature. I think in the next few years, right? And so then from the application of machine learning or computation side, I think it's going to go back to be getting a more systems level understanding, right? I mean, so when I was doing my PhD, when I was doing my postdoc, right, this word systems biology was really en vogue when everybody went to systems biology, right? This sort of died down a bit, right? And I think it's part of the reason was that the understanding we could get from building larger systems level models was just not there, right? It was just too early to do that, right? We didn't know what most of the proteins looked like. We didn't know how they interact, right? We didn't know the engineering relation really, right? So I think with so much great work going on now on sort of the wiring piece, on the gene relation piece, on the protein-protein interaction piece, right? I think soon it'll be time to do build system level models, but then for real, right? And,
yes.
things that actually have predictive power, right? Which we never could in like the next 2000s, right?
If I may add to that, so we already see how general AI models like language models and vision models and audio models are starting to integrate and converge right into one unified model or one interconnected model. So I see the same path will follow in our domain, right, where we connect these different layers of biology into a systemic model.
Yeah, I also agree, and I think this is a nice opportunity for our field because let's say in a wet lab environment, consider neuroscience, it is not easy to work both on gene regulation and on proteins and on cells and on electrophysiology, but also on brain regions, on the connectome and on MRI and EEG. It's nobody would be able to bridge these gaps experimentally. And so, but from the data and through models, I think this is, for a computational field, very interesting to actually start thinking about bridging these scales and do causal inferences over these scales, which an experimentalist has much more difficulty, I think, to actually tackle by an experiment. So this will be a really important phase for AI.
Yeah, this will be a lot of fun.
It will be a lot of fun, yeah.
I mean, just picking up on the data question, I suppose. I saw a comment on the Nobel the other day about how, essentially that was that was enabled by open, high quality, standardized data sets. Essentially, that was the point. And I think because Mariana, you mentioned as well the kind of the volume of data. And I suppose do you see is that question of the ongoing availability of data a kind of challenge? Are there challenges associated with data, or is the kind of application of machine learning, deep learning tools to data presenting new challenges? Do you think in terms of?
I mean, so I have one perspective on this now, having now actively worked in the biotech industry for a couple of years, is that there's a lot of data out there on different properties of antibodies or of drugs, right? And there's but the vast majority of the data sits in companies, right? And these companies have zero incentive to make it open. They have there's a lot of disincentive, right? Because there's IP considerations and all these things, right? And so there has been always efforts to try to get this data somehow unified and available and make it and then be able to train models, right? As far as I know, that hasn't really happened yet, right? Because there's so many hurdles, right? There's so many issues, right? Once you get to talk to these all these different players, right? But so that is so the simple issue of data availability is I think that is a huge issue. I think, and when you talk about stuff that's going to be commercially valuable, right? Which is a lot of it.
Yeah, I tend to agree with you. I mean, even though in public data sets we still have a large amount of data, some of them are very restricted to big pharma companies and so on. But maybe I would say that the biggest challenge here is lack of metadata and lack of homogenization of the data, because without metadata, the data sometimes doesn't make sense. And and it's, I know that for some metadata, we need to protect people's, like for sensitive data, we need to have this special care, how we share the data. But in our community, in general, people don't like, they kind of protect their data. They don't want to share this metadata. So this is a practice that I think we should change somehow. I don't know.
You opened a can of worms with the data question, you know. So I completely agree with you that the data is messy and it's noisy, and we invest, I think 80% of our time goes into data curation, just cleaning up data sets before we can model. But also to previous point, data is siloed in pharmaceutical companies, also in hospitals, especially in EU, where you have different regulations in different countries. It's so hard to get access to medical data. I can give you one example from Slovenia. So we have a lung clinic, right, dedicated lung clinic that treats all Slovenian lung cancer patients in one place. So they have this great data collection because homogeneous population, standardized practice, and so on. They collect RNA-seq, but it's so hard to get access to that data to the extent it doesn't benefit the patient anymore. So I understand those rules, right? GDPR and other European legislation is here to protect the patient, but at some point, patients are not getting the benefits if this data would be used for research.
Stein, did you want to comment on the data? Or you?
Well, just as an example I was thinking about today is that we've been usually doing model organism research and so on, but our recent investigations on the human genome have, of course, confronted us with questions about how much other data is available for use for training, and how do we share our data, and what do the models contain. So our models that I showed at the end of my talk were trained on human data, all post-mortem, so GDPR is not a problem there. But it's still sensitive, and we have gotten used to data, human sequence data from genomes, to protect it at EGA, to put under controlled access, so that people who want to use it can use it, but there's a data access agreement, and we all got used to that. And now comes the model era, where so we can put the raw data in protect it, but then release the model. But like Tim Hubbard was showing this morning, I thought it was very nice, I think. You cannot really tell what is in the model. Mm-hmm. The model may be able to regenerate, given a start of a personal genome, may be able to fill the rest in, and actually this way you can reconstruct somebody's personal genome. And so these things have not been solved yet, and now that we are starting also a new center, I'm thinking, if everybody has to solve these things by themselves, it's going to be a misery. So it's good that Tim and Elixir and start to work on this, but it would be great to have some more shared facilities in Europe, like what EBI is doing on data service, that there is additional guidance or infrastructure to make this happen. I think that is what we are now, at least what we are waiting for a little bit. So that we don't have to reinvent the responsible AI and data all individually. It will never work. So the field is going, the research is going faster, of course, than the infrastructure and then the legislation. So then.
No, it feels like there's always been a kind of arms race, in a sense, about the ability to de-identify data versus the ability to re-identify data. And it feels, after Tim's talk as well, that there's, the parameters of that race have shifted slightly? I mean, is it something that you're encountering as you're trying to do your work, that you're kind of the bounds of like when it is that you're have concerns about privacy or data disclosure are changing, or
No, yeah, I think this is something that we have to accommodate all the time. I just received, for instance, from one of our funders that I have to put some, like the consent, how do I say this, the consent that patients, they have to sign to participate in the research. I have to state a specific phrase there, and I have to now, store the data in some specific security requirements. So this, I think, as he said, I think we are learning the way, and we have to accommodate everything on demand, because although I think we can anticipate some problems, as we are saying here, things are happening so faster that it's hard to anticipate everything. So we have to adjust all the time, right?
I suppose actually to go back to the kind of like how you, where the kind of skills and resource comes from for that, I suppose, because you mentioned Elixir and other things. Do you feel that, you know, as researchers, often we say, researchers should be kind of responsible for all of this, you know. As an ethicist, I tend to kind of walk around a lot going, researchers need to be more responsible. But do you feel that there is that need for kind of new, so obviously there's a huge number of guidelines and guidance around trustworthy AI, responsible AI, but do you feel that there's kind of a gap around biology and around kind of biomedicine, or do you feel that we're just kind of refining? Is it a gap, or kind of is that gap closing, I suppose?
I think the gap is, I feel that there is a gap. If I compare with animal research, for example, we can do mouse experiments in the lab, and we send in, files to the ethical committee to perform these experiments. But as an experimentalist, we have to go through continuous training of the use and legislation of animal experiments. But that, and we hate that, right? It's like really misery to do that. But it's still there, and so we have always all these additional things we have to do as researchers that take us away from the daily research. It's true, but on the other hand, a minimal set of a minimal structure in our place is actually not really in place. So the ethical committees they don't know how to deal with AI models yet. There is no guidelines to follow for groups. You want to do AI research, then this is the way that you can send in your files. For ERC grants, we have to submit an ethical AI file as well, so that it is starting, and then makes you think about this. But in the institutes, I don't think this is really commonplace to have training and ethical committees being there to assist researchers in follow-up responsible AI. So for, at least in Belgium, this is not really completely implemented. While for animal research, it is really solid and like over the top almost. But yeah, so we may take examples, the good things there that might be useful, because it forms a guide. I think there's teaching, there's certificates that people get, to work with mice and so on. Not that we need that for AI, but there's a whole infrastructure and I think in the AI research there is almost nothing.
I completely agree with you, and I can share that in Brazil we are also discussing this, but there is no formal regulation about what to do, how to do. So, yeah, we are still talking.
Emerging area. I am going to open up in a minute to for kind of broader questions as well, rather than me hogging them all. But I wanted to come back actually to the one of the reasons that I co-opted you onto the panel, Mariana, was that because in your kind of conversation on ethics and as well, and I think a couple of other speakers mentioned questions of data bias and the kind of representativeness of data, and I suppose, I mean, and that is something that kind of comes up repeatedly as a question, and whether it's something that you feel that we're making progress on or finding better approaches to, or whether it's just something that, you know, is it something that people talk about and then kind of walk away from? And
yeah, I think we are more aware of this bias, but not necessarily tackling this bias, because for avoiding them we need representative, right, diversity, and as I mentioned, and now I'm talking specifically about omics data, most of people that are represented in those data banks, they are either Europeans or North Americans, so there are lots of populations that are not represented there. So if we are using those, this kind of data to build models and to represent our population, we are aware, of course, that we are not representing tons of people in the planet, right? Yeah, I think, as I said, I think we are more aware, but not necessarily tackling this problem. And another thing I see every time that someone's building a model, people are very concerned about what the model is doing, how the model is performing in terms of assertiveness, in terms of if the model is correct or not, but it's not taking consideration what the model is doing wrong. I mean, is this model, for instance, predicting, I don't know, not recognizing a specific face, or is this model not considering a specific ethnic group? So I think this should be an exercise when we are also building those models, to also pay attention to what we are skipping, what is missing, right? Not only what we are doing great, but also what we are missing.
And Miha, is that something that you...
Yeah, absolutely. So diversity is extremely important to us. We want to make sure our models perform well across diverse demographics. So we made it a standard practice to investigate model performance across these compounders like gender and ethnicity, and just the standard pipeline that we always look at that. And we invest a lot in forming partnership across the globe. So we have partners in East Asia, South Asia, Middle East, North America, but then we also realize we don't cover all ethnicities equally, and we are in discussions with cancer centers in underserved populations as well.
Yeah, we do, as molecular people, we don't deal with that a lot, right? I mean, we build protein models, right? So, and I mean, I suppose we could deal with it in the future in terms of, you know, targeting certain variants, right? But right now we target a target, right? And that's supposed to be universal.
It's interesting, kind of I suppose at what point, because obviously, and actually you've kind of almost sat in a kind of translational pathway, yeah, perhaps, by complete, but I suppose it's that question of when do all of these things that we put under this banner of responsible AI or ethical research, when do they become issues for different research areas at different times? I think that's a kind of, to me anyway, it's an interesting question. But I am going to open up to the room as well for questions. Apologies if I can't see you because the light is straight in my face. So, so if you're, but I think John,
So, like everyone else, you're suffering with data curation because no one wants to, and it's hard. But I don't work for EBI, so I'm just going to throw them under the bus. But if you had one sort of wish from EBI or a large data centre, I mean, A, do you think it's practical that they could provide data to cut down on that 80% time? And if so, what sort of thing do you want them to do?
Is that for anybody?
That's for anybody in the room.
Anybody want to volunteer before I just being aware that for the previous questions I've gone that way along the table. So
we think we can have standardised general curated data sets because each sample is different. You try and do different things. Would that even work?
I have no clue because then you could limitate creativity and new discoveries. On the other hand, you could facilitate one thing that. So yeah, maybe it could be like, maybe if someone wants to use the homogenized data, this would be helpful, but also have access to raw data in a controlled way would also help people who wants to be more creative and do different things.
Is it more useful? Because you get data from heterogeneous sites and bring it all together. Is that probably useful for someone to do that?
Absolutely. So I think there's extreme value in standardization of metadata, also of library prep, or the whole process from working with, from the sample to the data, right, in the lab. We've seen some great examples, and that is possible. Just if you look at the ensemble, right, they managed to standardize gene IDs and put all this effort under one roof. That's a fantastic effort. I think it will require a lot of more resources to standardize other data domains, but I think that's necessary, and that's how we should move in the future.
Yeah, I agree. CBio portal is a very good example of homogenizing data and fostering new discoveries. But this doesn't substitute the access to raw data, so then people can also apply their own pipelines.
Yeah, but the situation right now in the field is terrible. So I can tell you from the example in Slovenia, we are a small country with, a relatively small number of hospitals. All information systems for hospitals were designed by the same company, but all information systems in every hospital is different. So they would have different data schema in every hospital in Slovenia, although it was designed by the same company, and it's a fairly small country.
We have this issue in only one hospital.
Can I talk now? Okay,
oh, yeah,
cool.
Sorry.
I had a question on how we keep academia relevant. If you look at large industry models like LLMs, these are so expensive to train that basically they have to be done by very large organizations. And so how can academia remain in the game when this much money's at play? And how can academia remain flat and equal across countries and groups when it requires this much scale, and in biology in particular?
That's a great question.
Well, maybe quickly, I think it's a very relevant point, which has a lot of dimensions. Probably there are the huge models that are trained in big companies. Luckily, so far, still often find their way to academia, either because the output is reproduced, like now with AlphaFold 3. Luckily, academic groups, or the weights are released, or they leak, and lead to LoRa fine-tuning and so on in academia, which requires less computational resources. I was more worried maybe 2 years ago. I think academia is coping, but it is an important thing because our students and postdocs, or certainly students, they are trained in universities and academia. That's the principle of a PhD student, I think. And so if they work on problems which are like completely out of proportion compared to the companies where such students would be recruited, that creates like a weird situation. So I think merely we have to be able to give the students the capacity to work with the most relevant problems. I think we have to keep that in mind. And on the other hand, I also think that in Europe, there's not so many examples of these large models trained in academia, even though there are some resources, quite some resources in tier 0. So I wonder why this is not working well. So, right? So I think on the one hand, we can rely on the big models from companies, but on the other hand, we should also be able to train them. Even if they require a lot of resources, a couple of those models should be able to no, I think it's weird that we're not able to get there, even though in Europe we have a common supercomputer where I bet if you submit a project to get among all the GPUs, you could get it, but I don't see it happening a lot. So maybe we need a little more culture there.
Maybe I'll add my perspective.
From the US, from Canada.
From Canada, right? I mean, so first of all, if anybody has any GPUs to donate, we'll gladly take them. So I mean, I'm joking, of course, but I'm joking, but I'm not joking, right? But so in my academic lab, right, we're limited to the Canadian public resources, right? And so you can get, my students can get maybe 4 GPUs at a time to train a model, right? Which is already difficult, right? But I mean, I will say that AlphaFold 2, right, was presumably trained on something like 10-ish GPUs, 10 J100s or something like that. So the AlphaFold 2 training wasn't super expensive, right? AlphaFold 3 was probably trained on 500 or 1,000 GPUs, right? And so that's at a scale where it's I would say it's difficult to achieve as an academic, right? I mean, some people can make it, right? You can get donations or whatever, and you can get these resources. Some academics can do that, right? Most can't. We can't. But I will note that it's still several orders of magnitude away from like GPT-4 or GPT-5 or the really big large language models, right? Which are trained on tens of thousands, hundreds of thousands of GPUs, right? So that's when it's like utterly unachievable. When training this model costs you $100 million or something like that, right? Mm-hmm. So in biology, I think we're probably still, and I feel like we're gonna remain just because of data limitations. We're gonna remain in a world where you can get the resources if you really, really want to, and that if you have good funders, you can get the resources probably, right? But still, it would be nice to get. It would be nice to have more GPUs always.
So the general rule in like AI training is garbage in, garbage out. So how do we really address the reproducibility starting from labs? And how do you think that these new workflows and automation comes into play to solve these issues with reproducibility?
Do you want to start?
So that's definitely a big issue for us. We do enforce good machine learning practices to ensure reproducibility in our processing or analytics workflows. So when you take the same sample and run it through our models 2 times, we guarantee you get the same result of that prediction. But then we also need to ensure reproducibility in replicates and to be accepted for clinical trial use. For example, for our biomarker to be accepted for clinical trial use, we had to do the analytical validation, and we had to show that for the replicates of the same sample, right, we can reproduce and make the same predictions with certain accuracy. I guess at this level, right, when you want to enter the clinic, you need to show reproducibility.
I used to say the same phrase for my students all the time: garbage in, garbage out, because if you have like an input and this input is compatible, like it's in a good shape, then you are going to have an output, right? And the way I see this is that maybe first you have to be aware if the data you have it's capable of answering your hypothesis, your questions, right? Because none of some data then they can't answer what you are looking for. And second, I think you should have your hypothesis very clear and what you should expect as a result, and having a ground truth as well. It's a good way of certifying that the model is performing good thing, like what would be expected, right? And 3, I think external validation data sets are totally necessary, and people don't use this as frequent as they should. So.
For me, at the end, is still always the experiment to do. So if models can be compared and reproduced, external validation data, orthogonal data can be used, and then I think it's really interesting to do an actual experiment that is designed to challenge the model, and that usually gives for us a lot of confidence when these things then actually work, right? For the proteins as well. And if these design proteins actually work, it's like, okay, seems to be a pretty good model.
That's a good indication.
Yeah, pretty good indication, yeah.
Hello.
Question.
So I was just wondering, we've talked about explainable AI and responsible AI, and I wondered if you see them as two sides of the same coin. You know, is explainability necessary for responsibility in AI? And as we apply more scrutiny to AI models, are we going to need to be able to dig deeper in them? You know, so if you want to see biases in the models, I assume you'll need more and more intricate explainability. And if you want, and so I guess that links in with using more and more resources. If we can minimize the models in some way, then you'll need more explainable model methods to see what it is that you need to really get at in those models. That was for everyone, sorry.
Does anybody want to tackle that one? So, I mean, ultimately, so the kind of question of can you have responsible AI that isn't explainable? You know. Anybody want to tackle that one? God, Philip, you're looking.
I mean, from my perspective, I think at least from my perspective as a sort of in the narrow focus on the protein world, right, is the explainability and responsibility as sort of orthogonal dimensions, I think, right, because from our perspective, right, the responsible aspect comes in through applications, right, and you want to make sure, and I think I'm signatory on one of these declarations of trying to limit the proteins that we do to things that are helpful to humanity and not harmful, right? And so in that case, in that sense, it's orthogonal, right? So you can be fully non-explainable or very non-explainable, but still try to be responsible, right?
I mean, so moving kind of, I suppose, closer to kind of application and kind of regulatory context, I suppose. Miha, do you kind of, is in that context, is that link between responsibility and explainability more aligned?
I think it depends a lot on the context, like mentioned, right? I guess applications like ChatGPT don't need to explain the mechanics of their model. So what they do is they would present use cases, because I assume use cases are enough for their application. But in the context of patient selection and stratification, explainability is a big part of that. And we need to build models that are explainable. You also mentioned the point that there's a trade-off sometimes between model performance and model explainability. So usually complex models would perform better than simple models, but simple models would be easier to explain. So which model do you take, right? We try to look at this change in performance, and if there is no significant difference, we would always go with the simpler model that is easier to interpret.
I'm just thinking out loud that sometimes we tend to choose the simplest model because it's easier for us to understand, but maybe there are things that we don't understand that are very important, and then we are limiting, the power of the tool to our capacity, not the. So yeah, I don't know. I don't have an opinion on that. I mean, no, I do have. I think, well, we should be as transparent as we can, and I mean, sharing codes. I know that for industry it's something complicated because somehow they're protecting their know-how, but I don't know. I think when it's possible we should share codes, and also, when possible, use all the explainability to understand the code, and also because then we can understand, as I said, what we are missing. If we cannot understand what the machine is done, then we cannot see what's missing, right? And then we are going to assume that this is the right way of going, but not necessarily.
It's interesting. I know there's some evidence in terms of the acceptability of kind of data-driven decisions in other domains, where the kind of people are more willing to accept a wrong decision than one that can't be explained to them. It's a difficult trade-off.
And there was a study, a user experience study, that people are willing to wait longer if they see a progress bar. I think that kind of fits here.
Yeah, kind of transparency in that sense about what's going on, or the idea that something's going on. Here.
Hi, my question got slightly scooped, but I was going to say my second backup question was, I wonder what the panel's opinion is on following trends, especially in machine learning. There's things like LLMs and architectures that are now very optimized for these sort of inferences mean that everyone starts putting them into their applications. And when it comes to explainability, I wonder whether it's worth, let's say the panel's opinion, whether it's worth striking out into unknown directions or into architectures that aren't as popular, and how that trades off in terms of when you're working in research or when you're working in industry.
I didn't completely understand the question fully, so could you rephrase it a little bit?
I'll perhaps try again. But basically, there are very popular architectures like large language models and transformers, which are achieving very high performance but aren't necessarily very explainable or interpretable. And I'm wondering, when you're looking at it from different points of view, like in research or in industry, how much agency, I suppose, do you have to explore new areas, and is that worth it? Or is it best to follow the trend and integrate these existing architectures?
I mean, speaking as an academic, I think we should always explore new ideas and new things, right? I think that's kind of our job, right? And I will restate again something I just I was saying in my talk, right? Is that there were, the advent of diffusion in protein models, right? Was arguably transformative, right? I mean, there have been many generative, paradigms before diffusion, right? Like the GANs, or the VAEs, the autoencoders, right? And many people have played around with them, and we even in my lab we have done stuff on them, right? But it never really worked, right? Even though a fair bit of stuff got into it, right? But then they had a new thing that wasn't arguably not that different, right? But all of a sudden it's transformative, right? And so I think you can only do that if you start experimenting with this new things, right? I mean, there has been chatter for a while of things that are gonna supplant diffusion in the setting, or they're gonna supplant transformers again in the LLM setting, right? There's every year or so there's chatter about new stuff. It hasn't happened, right? Obviously, right? But I mean, it may happen, right? And so you can only do that if you keep experimenting, right? So.
Well, I think instead of going like surfing the wave, maybe you should choose the appropriate model for your data set. For instance, when I'm working with a very small data set, it's very hard to apply any neural network, for instance. So I think it's very cool to try new things, but maybe first you should identify models that suit better your data.
Yeah, so we have a few initiatives that to investigate methods you mentioned, like transformers, and we invest in that because those would allow certain kinds of applications that are not possible with the current technology, and we want to explore those. For example, digital patients or digital twins, right? If you have a generative model, you can give the model a clinical study parameters and ask the model to generate or simulate a patient cohort with such properties, right? Things like that. So we are definitely interested in such technologies, but whether or not those would be accepted in clinical practice or how we would present that to the public, I think we're still very far away from this stage.
Yeah, I think we've got time for probably one more question. Yeah.
Sorry.
It's sorted out between yourselves.
Very quick question. Like, just a follow-on on this. Like, there's so many foundational models, like so many trained on almost the same public data that's available. Do you think this trend is going to continue with us? Or do you think people are continuing to take it, moving to the next stage of like doing in silico, fine-tuning those models into their specific data sets and use cases and finding more valuable insights out of this?
In my opinion, you'll see that, so you'll have few companies that build foundation models, right? Or few groups that build foundation models, and everybody else will focus more on the application layer, on how to use these foundation models for their particular use cases. So if there was a foundation model of RNA-seq data, the kind of data we work with, I would happily take that foundation model and apply it to our specific needs. That's one reason, but maybe the second reason is also funding, one that we already mentioned. You can do only so much with limited resources, so.
I guess it's also hard to like kind of get like if you are a company like industry versus like academia, it's harder to like fundraise if you're just like fine tuning on existing models. So what are the incentives there? If are we moving into like, okay, like you mentioned earlier in your presentation, training those foundation models on public plus proprietary data, or just moving into like using those in a fine-tuned fashion?
I mean, maybe I'll get to say something about one of my pet peeves. Because foundation models, that's one of my pet peeves. I mean, I think foundation model, that's ultimately it's a marketing term, right? I mean, for foundation model, it's just a model that's been pre-trained. That's a pre-trained model, right? The entire concept of foundation models is nothing more than pre-training is useful, right? I mean, that's right. That's effectively what it is, right? So I think if you're ultimately, it's just a question of how much compute do you need to train a model from scratch, right? If that compute is very large, if that engineering is very large, right? Like, I mean, undoubtedly building a GPT is tons of very smart engineering, tons of compute, right? Rebuilding that is going to be very, very difficult, right? But on most data sets that we deal with in the bio realm, right? Training a protein language model, right? That's with billions of parameters. That you can do that. You can do that. With a reasonable amount of resource, right? If you just put your mind to it, right? That's why I think. Anyway, so that's just my pet peeve.
Okay. Who would like the last word on this? Okay. Or not? Okay, well, in which case, I'm going to say, just join me in saying huge thanks to the panel for wrapping up a great day. So thank you.
Thank you. Thank you.